All right, today we are actually on page 37 in your notes, and we are going to talk about nucleic acids and carbohydrates today. But just before I move on, the activity that you already did today was over protein and protein folding. And so I just wanted to show you that it's not just temperature, chemicals, and pH that can cause a protein to become misfolded. Genetic mutations are going to also result in a misfolded protein. And so here we have the primary, secondary, and then it skips primary, secondary, tertiary, and then quaternary structures of normal hemoglobin. And remember, hemoglobin is going to be in red blood cells, going to help carry oxygen. Down here, you have a genetic mutation that results in a messed up amino acid, a wrong amino acid. And remember, it's the interactions between amino acids that cause proteins to fold correctly. And if you have a wrong amino acid, the wrong interactions occur, the wrong folding occurs. And so a change in a protein structure is going to ultimately change its function. And so with sickle cell anemia, there is a genetic mutation that causes a valine to take the spot of a glutamine. And so that causes a misfolded hemoglobin, and then that causes a misshapen red blood cell and actually a sickled shape. And so that can result in blood clots, heart issues, fatigue, and so a lot of problems. So on page 36 at the bottom where it says change in structure, you're going to want to say a change in structure can change its function. I'm just going to skip over that. And then today we're going to, like I said, cover nucleic acids and carbohydrates. So the job of nucleic acids are to store hereditary info. And so you have a table here where you're going to compare DNA and RNA. And what you're going to want to do, I'm going to read through these real quick and then you're going to want to pause. So DNA is double-stranded, RNA is single-stranded. DNA's nitrogenous bases are A, G, C, and thymine, T. The nitrogenous bases for RNA are A, G, C, and uracil. So remember, RNA has uracil instead of thymine. It has a U instead of a T. DNA stores hereditary info. RNA carries hereditary info from DNA to ribosomes. And then the sugar that that a nucleotide for DNA has is deoxyribose, and then the sugar for RNA is ribose. So you're going to want to pause and write these things down because these are going to be important to remember. And then when we talk about DNA replication and then going from DNA to RNA to protein, we are going to revisit all of this information again. I just want you to know the basic information about the components that make up nucleotides and nucleic acids. So DNA and RNA are nucleic acids. Nucleic acids are made up of nucleotides. So the nucleotide is the monomer of a nucleic acid. And a nucleotide is going to consist of three parts: a sugar, a phosphate, and a nitrogenous base. And so right here you have the phosphate, it's the circle. Right here you have the sugar, it's the pentagon. And then right here you have your nitrogen bases. So you're either going to have your purine or your pyrimidine nitrogen base. And remember, the nitrogen bases are going to be either A, T, C, G, or U instead of T if you're talking about RNA. All right, so these are nucleotides linked together right here. And then it kind of zooms in to show you a little bit more detail of a nucleotide. So a phosphate, a sugar, and a nitrogenous base or a nitrogen base. And then those bases are here. This is just showing you the different sugars. So DNA has deoxyribose, RNA has ribose. I don't need you to know the exact differences. I just need you to know that DNA has deoxyribose as a sugar, RNA has the ribose. And then you have the phosphate and then the nitrogen base. The nitrogen base, A pairs with T, C pairs with G. Unless you have RNA, A from DNA is going to match up with a U for RNA. All right, and so these are showing you your nitrogenous bases. These are what are called pyrimidines. They're single rings: cytosine, thymine, and uracil are pyrimidines. And then purines are going to have two fused rings. Adenine and guanine are your purines. And then this is showing you once again the sugar, deoxyribose, and ribose. And like I said, I don't need you to know this little like small detail that deoxyribose has an H instead of an OH, but it is kind of it is a pretty subtle difference. I mean, a really small difference, but it is a difference. I do need you to know what the nitrogenous bases are. I need you to know that adenine and thymine pair and that guanine and cytosine pair when it comes to DNA, and that adenine is going to pair with uracil of RNA, and that RNA has a uracil and not a thymine. And so this information is going to become very important when we talk about the synthesis of proteins. And so DNA is here in the nucleus. DNA is going to undergo the process of transcription. Transcription is going to make a single-stranded RNA. The single-stranded RNA is going to leave the nucleus through a pore. It's going to go to a ribosome, and then it's going to undergo the process of translation, where tRNA is going to bring in amino acids, and a polypeptide is going to get formed. And then that polypeptide, remember, is going to enter into a chaperonin, and then it's going to get folded. So we are going to just stop there with nucleic acids, and I will go back to that later. I want to talk a little bit about carbohydrates next. And so carbohydrates are going to serve the purpose of fuel and building material. What I want you to write on top of fuel is quick energy, quick energy. And then building material, they can come in the form of simple sugars like fructose or glucose or lactose, or they could come as really large polymers like starch and glycogen, and I'm going to show you all of these things that I'm mentioning in just a minute. The way you're going to recognize that you have a carbohydrate is you're going to have carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. So carbon, hydrogen, oxygen in a 1 to 2 to 1 ratio. So when you see a molecule, you're going to know, yeah, that's definitely a carbohydrate. The monomer of carbohydrates are called monosaccharides. When you have two monosaccharides covalently bonded together, that's called a disaccharide. And then when you have multiple long chains of monosaccharides, that is going to make up a polysaccharide. And so here are some examples of monosaccharides, also known as the monomers. And so this is where you're going to start writing in your notes. So monosaccharides are the monomers. An example of a monosaccharide. So when you see one ring, and when I say ring, I'm meaning either like a pentagon or a hexagon. I'm talking about a monosaccharide. So glucose and ribose are examples of monosaccharides. Polysaccharides, it's really important to know all of these examples, and I'm going to go through some pictures in just a minute. A polysaccharide can function as for the purpose of storage. So in plants, starch is going to be the storage carbohydrate. And so you're going to find a lot of starch in things like potatoes. So that's going to be the storage carbohydrate for plants. In animals, the storage carbohydrate is glycogen. And what we do is we store glycogen in our liver so that if our blood glucose levels get too low, our body can access glycogen, break it up into little glucose pieces, and then release it into the blood. The other polysaccharides, there's actually 4 polysaccharides you need to know for me. So you need to know starch is for storage in plants, and it's a really large molecule. Glycogen is for storage in animals. Cellulose is for structure in plants, and then chitin is for structure in arthropods. So pause if you don't have that written down because I have something really, really important that I need to say. Okay, so once you have that written down, what I need you to write underneath is all 4 of these are made up of many, many, many glucose molecules. They're all made up of a bunch of glucose molecules bonded together. Remember, glucose is C6H12O6. It's a hexagon. You should have seen it plenty by now in cell bio at least. So these are all large polysaccharides all made up of glucose. So what's the difference? If they're all big, long chains of glucose molecules, what's the difference? The difference is the way that the glucose molecules are bonded together. Okay, and so Carbohydrates are going to have the functions of energy storage, quick energy, and cell signaling. So I'm kind of moving on. I'm under polysaccharide. I'm moving on to common monosaccharides. Then I'm going to talk about synthesis, and then I'm going to come back to those polysaccharides once again. So that was just a broad overview of what I'm about to talk about. Talking about the functions, then I'm going to talk about monosaccharides again, then I'm going to talk about polysaccharides again. So I would write this down. The functions of carbohydrates are for energy storage, quick energy, and cell signaling. Here's the structure and the classification of some common monosaccharides. I want you to notice that glucose, galactose, and fructose are all C6H12O6. They're all monosaccharides. So then what's the difference? It's just the way the atoms are arranged. So they're very similar. They're all the same chemical formula, but the way the atoms are arranged are just different. All right, so I'd like for you to know glucose, galactose, and fructose all have this formula, but because of their arrangement of the atoms, they are different. They're considered different molecules. Okay. All right, so glucose is actually a linear molecule, but it will fold into a ring when it's in an aqueous environment. So that's why you're going to always see carbohydrates as rings, because in the cell, they're always going to be in an aqueous environment. Okay, this is going to show you the process of carbohydrate synthesis, and I want you to notice it's actually very, very similar to what we talked about before. So right here you have one glucose ring and another glucose ring. The OH of one glucose and the H of another glucose combine to release water, and then a bond is formed. What I need you to know is the covalent bond between monosaccharides is called a glycosidic linkage. So the covalent bond between monosaccharides is called a glycosidic linkage. I would circle that word. So remember, the covalent bond between amino acids is called a peptide bond. It's the same thing. The bond between two monosaccharides is called a glycosidic linkage, and it is achieved through this dehydration reaction that we saw before. And then two glucose molecules bonded together makes up what's called maltose. It's like malt sugar. All right. Glucose bonded to fructose. Same thing. A water molecule is released and a glycosidic linkage is formed. Glucose bonded to fructose is going to make the disaccharide called sucrose, which is table sugar. All right. I'm sorry if you can hear my kids in the background. There's no way I can get away from that. God, they're so loud. All right. So those are your monosaccharides. So I gave you some examples of monosaccharides right here. Here are two examples of disaccharides. Maltose and sucrose are disaccharides. There's two rings bonded together. Now let's talk a little bit more about your polysaccharides, okay? Let's talk about cellulose, starch, glycogen, and chitin. So I'm on page 39 now. Let's talk about cellulose versus starch. So cellulose and starch are both found in plants. Okay, and cellulose and starch are both large polysaccharides made up of glucose molecules bonded together. You have what's called an alpha glucose, and then you have what's called a beta glucose. And alpha glucose is going to have this hydroxyl hanging off of the number one carbon kind of downward like that. And then the beta glucose is going to have a hydroxyl that's up. All right, so you can see the difference. Here's an alpha glucose with the hydroxyl kind of hanging down. Here's a beta glucose with the hydroxyl group up. All this means is it's still glucose, but the hydroxyl groups are in a different position. And so why does this matter? Well, because of the position, the spatial orientation of those hydroxyl groups, alpha glucose monomers are going to bond together differently than beta glucose monomers will. And so because of the different bonding that occurs between them, starch is either going to be able to kind of form a little helix, or it's going to form branches. And then cellulose is going to form sheets. And so you can see right here that here is a chain of glucose, beta glucose monomers bonded together. Here's another chain of beta glucose molecules bonded together. And then there's hydrogen bonding between them, and so they form in sheets. This is what makes cellulose so difficult to digest. And this is what makes cellulose, when you hear about eating fiber, people are actually talking about cellulose. It draws water into the gut because you don't have the enzymes as a human to break bonds, to break these beta glycosidic linkage bonds. And so it brings water into the gut, and then it actually just is supposed to like keep you regular. And I know that's gross, but that is a biological process. So eating cellulose is very important as a human. All right, this picture is just showing you plant starch. Remember, starch is made up of those alpha glucose monomers. And so because of the way they're bonded, they don't form sheets. They can form kind of like these spirals right here, which is amylose, is a type of starch. Or it could form these just a few branches. Amylopectin is found in plants, also as a storage carbohydrate. For you have glycogen as an animal. We have glycogen, which is very similar to starch. So starch is the storage polysaccharide in plants. The storage polysaccharide in animals is called glycogen, and it's very similar to starch, except it's more branching. So I would just write down next to glycogen, more branching. And so these are alpha glucose monomers right here, so they can form a bunch of branches, and they aren't in sheets or anything like that. And so glycogen is what you store in your liver as your as your reserve for when your blood glucose gets too low. And then lastly, chitin. Obviously, chitin is found in animals, and it's found as the structural polysaccharide in animals. So the structural polysaccharide in plants is cellulose, and then the structural polysaccharide that's made up of monomers of glucose bonded together would be chitin. And so you have your four major polysaccharides. You have starch, glycogen, cellulose, and chitin as your large polysaccharides. Remember, they're all made up of glucose monomers bonded together. The way that they're bonded together is going to give them their unique properties. And then we talked about a couple of disaccharides, maltose and sucrose. So I'm just kind of reviewing backwards right now. And we talked about the bond between monosaccharides is called a glycosidic linkage. And then we talked about some monosaccharides like fructose, galactose, and glucose. And then if I would like for you just to go back and review the notes and to look at the dehydration reaction that occurs and how that forms a glycosidic linkage. And so at this point, we've talked about carbohydrates serving the purpose of quick energy. So your monosaccharide, like glucose, is going to serve as quick energy. And then we talked about carbohydrates functioning for structure, and then energy storage. And then the last thing that I had said was cell signaling. Cell signaling and cell recognition is actually an important function of carbohydrates. And so our example here shows your blood types. And so this blue one right here is a certain carbohydrate that's designated as the type A. The green with the arrow is designated the type B carbohydrate. And then when you have both of them together, you would have AB blood type. So this just serves the purpose of cell recognition. So this is why if a person has B blood type, you see how they have the green arrows? They have a particular carbohydrate that's hanging off of the cell membrane of red blood cells. If they were infused with the blood type of someone with type A, their body would reject it because it doesn't recognize it. So your cells are really good at recognizing self from non-self based on the carbohydrates that are hanging off of them. And so I don't need you to know blood type or really understand it. It just depends on who your cell biology teacher was. I kind of went into it in my class, but we will touch on blood type at some point in the future when we do genetics. But I just wanted you to know that your blood type is based off of a carbohydrate chain that is hanging off. And so what I'd like to do is stop right there. I am going to cover lipids in the next lecture, which is actually pretty quick. And then we're going to fill out, or I will assist you in filling out that macromolecule chart as well. And then that will be the end of the biochemistry unit. It's really a quick unit. It'll be our quickest one. And so the next time I have a lecture, that will be the end of our material for your test. And I will see you guys later.